Okay, anomers. So I had done that like sort of experiment in class there with you guys last time to get you to understand stereochemistry and particularly the interconversion around this aldehyde group here in glucose, and how the glucose can interchange between an open chain form and the closed chain form, depending upon where this hydroxyl group down here on this carbon, which side of the carbonyl group it attacks. You're going to end up with either alpha glucose or beta glucose. And I want you to understand that distinction. It's called the configuration around that carbon. That distinction between alpha and beta glucose has consequences in terms of the kinds of polymers we're going to be able to make out of that glucose. So starch, for example, is made out of alpha glucose, and beta glucans make polymers that tend to be resistant to digestion. So like wood fiber or chitin, the shells of shellfish, are made out of these beta glucans that are resistant to digestion. A lot of fiber has beta glucans, whereas alpha glucans are things that our body is able to process as energy. All right. So yeah, this whole process of going between alpha and beta glucose, it's called mutarotation. I guess that's not especially important in terms of food science, but it's simply a reflection of how changing the configuration about that carbon changes the optical rotation. We talked about optical rotation last time in class, how the stereochemistry of a molecule is going to bend light polarized light in one way or another. You guys remember that? Okay. Just a little bit of visual semantics here so that when I put all these structures of sugars up here, you don't get lost. There are really a number of different ways that scientists have tried to depict sugars on the two-dimensional plane. So what you're looking at here are 4 different ways to depict the exact same molecule. It's the exact same molecule, beta-D-glucose. So we talked about the Fisher projection. You can also use the Fisher projection to show the closed ring form, but with this weird box over here. I'm not going to go into the history of all these, but there's a Mills projection where the dotted line means the hydroxyl group is going away from you into the plane, and the solid line means it's coming up out of the plane towards you. Likewise, this is below the ring, above the ring, etc. And this gives like this chair conformation thing gives a better depiction of the actual bond angles that we're talking about. In all cases, the CH bonds, the H of the CH bonds have been removed. There's actually a hydrogen sticking up right here, for example, and there's going to be one sticking out right here, for example. All right. Yeah, so just some visual language around how to look at sugars on a diagram. Yeah, so we've talked about enantiomers. Enantiomers are non-superimposable mirror images. So D-glucose, which is also called dextrose, it has an enantiomer, a non-superimposable mirror image known as L-glucose. And you see the picture of it right there as if it were reflected, either open chain or in the ring form, reflected in some mirrors. And why do I bring this up? Well, it's because D and L glucose have almost the same physical and chemical properties. So when I may say physical, they have the same melting point. They have the same If you were to burn them, they would have the same caloric content, right? If you were to, they have the same solubility in water, right? They have their, in terms of their physical and chemical properties, they are identical to one another. However, one of them, the body can utilize, and it's like the currency of life, and the other is like from a foreign planet. Biology doesn't really know what to do with it and doesn't utilize it. Why is this? Well, it's because sugars are often, like the biochemical transformations that happen to sugars are mediated by enzymes, big protein enzymes that have some three-dimensional structure that are going to recognize the three-dimensional structure of the sugar. So here's an enzyme, a depiction of an enzyme in orange here, and we see D-glucose fitting into the enzyme pocket. Whereas if we try to fit L-glucose in there, it's not going to at all. This hydroxyl group is out of place, this one's out of place, this is just ramming into the structure of the enzyme. That doesn't fit at all. So how do we think about that? Imagine the protein is like a glove, right? And you can slide your hand into the glove if it's D-glucose. Whereas if you have that same protein, if you have L-glucose, you cannot slip your right hand into a left-handed glove. All right? Does that make sense? Okay. This is weird. This is a weird fact. Again, when we're talking about D and L, we're just talking about the configuration, about this fifth carbon in a hexose. The one that's the hydroxyl group is going to react with the double bond O to form the ring. The configuration about that carbon in just about all sugars in nature, which I told you is 85% of the biosphere is made up of some kind of sugar. Just about all sugars on Earth are D in structure. All right? Just about all of them are D in structure. And a weird thing is that almost all amino acids, in fact, all amino acids of the 20 that life is made of are L. We call it L because it bends light to the left. It's not actually, you can't, it's comparing apples and oranges, the D and the L between sugars and amino acids. But all life has L amino acids and D sugars. And you may say, like, so what? Well, why is that? Why is it not a mix? Or why isn't it all L sugars and D amino acids? People have pondered this a lot, the kind of people that think about where life came from and how all the amino acids and sugars started on Earth. And one of the things that the most compelling argument is that there is some background radiation in the universe. This is some pretty far out stuff. There's background radiation out in outer space that is polarized light. It's circularly polarized that has a particular handedness. And they say that it's related to the twist in the universe when the Big Bang happened. The Big Bang did not happen uniformly. That light, there was a little bit of a twist to it, and light got twisted in a certain direction. They call this the Lyman frequency, and the circularly polarized background radiation in the universe gave a slight advantage to certain enantiomers, chemical enantiomers, as the chemical economy was growing prebiotically on Earth before biology happened. Yeah. That's a little weird factoid. So there could have been life with all L sugars and D amino acids. And it would have been this bizarro mirror image world where everything was the non-superimposable mirror image of life on Earth. All the chemistry, all the biochemistry that happens would be the same, but not able to interact because it would be this chemical mirror image of what's happening on Earth. it's too interesting to not go there. I wanted to share that one. Okay. Are there any questions on monosaccharides? Because I'm going to move on. I'm not going to talk about glycemic indexes and all that yet. I'm going to save that for metabolism. And I just want to get the nomenclature of sugars down first, what we're actually talking about before I talk about what they do. So disaccharides. We can take these monosaccharides and build them up into more complex structures. And the first way that we do that is by creating disaccharides. We create a disaccharide by building what's called a glycosidic linkage through what the type of reaction is called a dehydration reaction. So I have these simplified sugars here. I've stripped everything off but the action for simplicity's sake, so you don't get confused. And I'm just showing the atoms that are going to be involved. We have two hydroxyl groups, and typically one of the hydroxyl groups, the one that has both the O and the H highlighted, that O and H is typically on the anomeric carbon. That's the carbon that has the double bond O, the aldehyde or ketone. All right? That hydroxyl group is going to interact with some other hydroxyl group on another sugar. Water is going to pull out of there, and so this is going to be called the non-reducing end. This is going to be called the reducing end. Don't worry about what that means. It just gives a directionality to it. This reducing end sugar, that oxygen is going to form this glycosidic linkage, or an ether linkage is chemically how you would describe that. This is now a disaccharide. Yeah? Yeah, so, yes, that's right. Okay, and they call it a condensation or a dehydration because what we're doing is pulling out water. Water is one of the products of this reaction. We can go in the opposite direction and go from a disaccharide down to two monosaccharides, and instead of a condensation reaction or a dehydration reaction, you call this a hydrolysis. You're using water to hydrolyze, cut, water cutting reaction. You're just going to add water across this bond and get us back there. So this arrow is actually in equilibrium. The arrow points in both directions. Yeah. All right. But we're only going in one direction, which is forward right now. So here's an example of one of these reactions. We can take two molecules of alpha glucose. And remember I told you alpha glucose is the sugar that makes polymers that the body likes to eat. Biology is able to consume, right? And can get used for energetic needs because it's easy to hydrolyze alpha linkages. Here is alpha glucose and another molecule of alpha glucose. We do the condensation reaction or dehydration, and we get an alpha 1,4 linkage. All right. So you see how this is carbon 1, 2, 3, 4, 5, 6. Carbon 1, and it's an alpha, and it's linked to the 4 position on this sugar. So this is carbon 1. You can always find carbon 1 because it is the carbon that is attached to two oxygens, right? It's either in the ring form, got two single bonds to two single oxygen, or in the open chain, it's going to have a double bond to a single oxygen. But it's always got two bonds to oxygen. So that's carbon 1, 2, 3, 4. This is an alpha 1,4 linkage. A lot of the chemistry, a lot of the biochemistry, a lot of the nutritional features of a particular sugar it's all about this linkage. It's all about the linkage. What kind of linkage? Because different enzymes are going to be able to recognize specific linkages and break them apart or form them, right? And so we're going to see some examples of that moving forward. Like we're going to relate it to lactose intolerance. We're going to relate it to how our body breaks down table sugar. We're going to relate it to how babies utilize mother's milk. All right. So yeah, this is maltose. Maltose is a disaccharide that they use it for a number of things. For example, if you ever had a malted milk, you can get maltose in a powder form that they put in malted milks or in like milk duds. Those things have maltose in it. That like chalky, kind of sweet, kind of odd flavor. That's the flavor of maltose. We can go taste it in the lab. Maybe on Friday I'll pull some out. And it's also used in brewing. So a lot of brewers will dump maltose into their beer because the yeast really like to use maltose. Huh? Yep, they eat it. Okay, so here's a different disaccharide called cellobiose, also made out of 2 molecules of glucose, except they are beta-glucose. And we can see now that this anomeric carbon has the hydroxyl on the other side. It's going to interact with the carbon 4 just the same, but now this is a beta 1,4 linkage. Beta sticking up, a 1,4 linkage, a beta 1,4 linkage. That gives us a disaccharide that is identical to maltose in every way except the configuration about that carbon. It's a super picky little thing, but it takes the disaccharide from being this delicious disaccharide that goes in milkshakes and beer to being something that will actually give you the runs if you eat very much of it. It is completely unusable by the body. It's called cellobiose. All right? Yeah, I'll give you a stomach ache. All right. So here are a couple common disaccharides. You know, there are literally thousands of different disaccharides that I could potentially throw up on this board for you guys. But here are just 2 very common ones. Here is, and I'm sure everybody has come in contact with both of these in their life. Lactose or milk sugar, even if you're lactose intolerant, chances were high you had it as a child. Is anybody lactose intolerant here? No? Well, then we've all had lactose, I'm sure. This is a beta 1,4 linkage, and I said earlier that these beta glucans are not utilizable by the body. What? But this is a beta 1,4 linkage, except that now this is galactose, where the hydroxyl group around C4 has a different configuration about that carbon right there. Has a different configuration about that carbon. So the enzyme that's going to be able to break that down is able to recognize lactose, whereas it wouldn't have been able to recognize cellobiose. So cellobiose looks exactly like this, except this carbon right here, the hydroxyl group, has a different configuration, lactose. So yeah, we don't have any people who are lactose intolerant here, but if we did, and you took some lactose, and you knew what that felt like, that's what it would feel like to everyone in the class if you ate some cellobiose. All right, it would give you diarrhea, bloating, stomach cramps, that kind of thing. That's what people with lactose intolerance deal with. Yeah, and I do have a molecular model. That's what all these links keep popping up for, I guess. You want to see a structure of it? I guess we, oops, no, come on back. Wow, let's keep going. So another disaccharide that I'm sure we're all familiar with is just table sugar or white sugar. You guys have all seen this before, obviously. And this is a little bit different because now it has a linkage between glucose, and it's an alpha glucose that's alpha 1,2 linked to the anomeric carbon in fructose, which is a ketose. You'll notice that this carbon here, which is attached to the 2 oxygens, is also attached to 2 carbons, which makes that a ketose, not an aldose. So sucrose is a polymer of glucose and fructose put together as a disaccharide. Right? I'm going to talk a little bit about these two these two disaccharides and how the body deals with them one at a time. First, let's deal with lactose. So there is an enzyme that the body has called lactase. Now I'm going to go through this, and there's a bunch of words that sound similar. In fact, one of the groups was talking about ice cream, and they talked about this a little bit. I think it was the ice cream group. No, it was the alternative milk group had been talking a little bit about this, and they were getting in their draft just mired a little bit in all these different words that sound similar. There's lactose, there's lactase, there's lactate, and lactic acid. Lactate and lactic acid are completely different. So we're just going to talk about lactate and lactic acid is what builds up. It's a metabolic byproduct of your cells. Lactose is this disaccharide, and lactase is the enzyme that breaks it down. Lactase, oh my God, come on back. How did I do that? Come on back. Lactase is this enzyme also known as beta-galactosidase. Breaking that word down, sidase means it's going to cleave a glycosidic bond, or that bond that bridges two monosaccharides, and one of them, the non-reducing end of that disaccharide, has got to be a beta-galactose. So it recognizes beta-galactose containing disaccharides and cleaves that linkage. It clips beta-galactose off. So this is how it works. We have the enzyme, the lactase enzyme, and the lactose comes and binds the lactase enzyme. We see a ribbon diagram of the crystal structure of it on the right-hand side. I believe right over here you can see the active site. This is the disaccharide in the active site. It's the enzyme sort of bends it and makes it the glycosidic bond particularly available to water to come in and hydrolyze that bond, and it can break it into glucose and galactose. Once that glycosidic bond has been broken, then the protein the enzyme releases it, and it's able to go in a cycle again. So enzymes do this process of catalysis. Catalysts are simply things that help a reaction go on, and they recycle themselves over and over and over again. So this is how, for example, babies or any of us in here that are able to drink milk, when you drink milk, the lactose that's in it, your body makes the lactase or beta-galactosidase enzyme that breaks the milk up into galactose and glucose. And then the body can absorb the galactose and glucose and use them for energy and whatever else it's using them for. Straightforward? Yeah? Okay. So here is an interesting model. So this was a genetic. This is based upon genetic screening of all of these Europeans and their ancestral origins, tracing the occurrence of lactase persistence. So prior to about 10,000 years ago, everyone on Earth at about the age of 5 stopped making the lactase enzyme. Your body just didn't make it anymore. All right? You didn't need to make an enzyme when you weren't drinking milk. This was before humans really had started, domesticating cows and drinking the milk of animals. So the only milk that humans were coming in contact with was human mother's milk, and that stopped at around no later than the age 5 or something like that. All right? So you just didn't need to make the protein. No point in making a protein you're not using. So prior to about 10,000 years ago, everybody on Earth was lactose intolerant. Being lactose intolerant is not like some weird mutation that makes those people strange, right? When I look around the room here, everybody in this room has more or less European origin in them. meaning that if nobody here is lactose intolerant, all of us have a gene in us that originated at that X right there in northern Germany. So about 9,000 years ago, there was a mutation that happened in northern Germany where there was the domestication of animals. They must have been drinking cow's milk into adulthood, and it was an advantage to be able to digest it as an adult. And so that gene spread throughout northern Europe, and you can see here how it spread through the Saxon invasion of Brittany and Ireland, not Brittany, Great Britain and Ireland, and has spread throughout people of European descent. Many Asian people are lactose intolerant because they have not come in contact with the lactase persistence gene. This is not the only place that this has arisen. It also arose orthogonally, right? So not related to this in Africa. So there are many Africans who are also able to drink milk, but a lot of Asians are not able to. I don't know. Is there anybody have an experience in their family of someone that is? Yeah, you have someone in your family that is lactose intolerant. My daughter is. She just developed that in the last 6 months. But not being able to drink lactose may put you out of step with your peers who are really into ice cream or something, but not completely, because you can go over the counter and buy supplements that are basically pills that have lactase in them. You can buy supplements and you just take the pill with the dairy, and the enzyme breaks down the lactose as you're consuming it. There's also dairy products where the lactose itself has been boiled, has been removed or broken down, treated. A lot of people actually prefer lactose-free milk because they say it tastes sweeter or better or whatever, because the lactose has been broken down to glucose and galactose. So it will change the flavor profile a little bit. All right, any questions on that? No? Okay, so now let's talk about lactose and mother's milk. The reason lactose is there, let's think about this a little bit. So the mother feeds her child, and about 7% of the solids in that milk, the milk solids, are lactose. So a large percentage of the carbohydrates, the bulk of the carbohydrates, and a large percentage of the total solids, 7%, is lactose. There is about 0.03 to 0.09%, and so a very small, like 1/10,000th of the mass of the dry mass of the milk is a different disaccharide called allolactose. I told you lactose was this beta-1,4 linkage. Allolactose is a beta-1,6 linkage. Same monosaccharides, galactose linked to glucose. Different linkage, a little bit of something called allolactose. I'm going to see why that's important in a moment. But first, back to the lactose. So these bacteria that live in the baby's gut, when a baby is first born, or in utero at least, its GI tract is sterile. There's no bacteria in the GI tract. And then the baby is born, and it comes through the birth canal and gets inoculated coming through the perineum of the mother. Even now, babies who are delivered C-section, often those obstetricians will take perineal swabs from the mother and inoculate the baby with that to be able to give them the right kind of gut flora start. So a huge percentage of the lactose that's in the milk gets taken up by those coliform E. coli bacteria that are in the new baby's gut. And this is what happens. This is what they do with it. So to begin with, let's say the baby has not fed yet. Here's a baby. It's thinking about dinner. It's looking, but it hasn't eaten yet. That E. coli, that bacteria, has this gene in it called the lac operon. It's a bunch of DNA, and the way the DNA works, there is this protein that attaches to the DNA called RNA polymerase. It's an enzyme that is going to transcribe the DNA and make RNA off of it, throwing your mind back to the genetic paradigm. That RNA is going to be used to make proteins that the cell is going to use, etc. So normally when there's no food in the baby's gut, there is this repressor, this red protein here, that plugs in, and attaches to the DNA of the lac operon and prevents the RNA polymerase from transcribing these genes here. All right? But now the baby has some milk, and it comes in contact with, the first thing it has to come in contact with is allolactose. The bacteria cannot absorb the lactose. It can't do anything with it. It's just sitting there swimming in the lactose. But it can absorb allolactose. That allolactose binds to this repressor. The allolactose is called an inducer. When it binds to it, it inactivates that repressor, and then now the polymerase can begin transcribing the lac operon gene. All right, so it's going to move its way along the DNA. It's going to make some RNA that's going to lead to proteins. First of all, permease. This is a membrane-bound transporter so that the bacteria can now take up the lactose. It's going to produce a lactase gene, a beta-galactosidase gene, that's going to be able to break down lactose and allolactose into the galactose and glucose monomers, etc. All right, and this was the first example of gene regulation that was ever identified. All right, it was the first example of gene regulation that was ever identified. Specifically, it was a negative gene regulation. The operon, which is the name of that gene, it's an operational unit of DNA where all the genes work together to achieve a single purpose. The operon is inactivated by the repressor that gets removed by the presence of the inducer, which is the sugar allolactose. These three guys here, Jakob, Monod, and Louf, received the Nobel Prize for that. What do we get out of it? These bacteria are eating all of our mother's milk. Why is that good for us? Has anybody ever had to change a new baby's diaper? Nobody? Okay, well, that's something fun that some of you can look forward to. The color of new baby's poop is, Ben, I'm sorry to do this to you, it's about the color of your coat. It's like this yellow mustardy color. It's like an orange, it kind of looks like some kind of a curry or something. It's the color of the Nobel Prize there. And the reason for that, has anybody ever had like one of those Adwala juices that has a lot of blueberries in it or whatever? Or taken a multivitamin with a bunch of B vitamins in it? What does that do to you? Turns your yellow crazy dark. Your urine crazy dark yellow, right? It's an overabundance of vitamin B. So baby poop is that color because those bacteria, the lac operon, the E. coli bacteria that are absorbing the lactose, they are making the B complex vitamins that are so essential for the baby's neurological growth. All right, because the mother's milk doesn't have that much B vitamin in it actually, and the baby needs the B vitamin. It gets it from the E. coli bacteria. For the E. coli bacteria to do it, they need to get the lac operon turned on. To do that, they need to get allolactose. This, what I'm giving you, is at the heart of the real difference between mother's milk and Similac. There are babies, like artificial milk is lovely. It's great that it's out there, particularly for mothers who choose not to or cannot breastfeed. But there is a distinct nutritional difference between mother's milk and Similac and that kind of thing, because Similac has all the lactose, but it does not include allolactose. Allolactose is super hard to synthesize, and it's extremely hard to isolate. And it's cost prohibitive for including it in prepared milk. And if there's not the allolactose, which is the inducer of the lac operon, then those E. coli bacteria aren't able to operate with quite the same efficiency that they would otherwise. I have a little bit of experience talking about how difficult it is to synthesize allolactose. I actually spent 5 years of my life as one of the projects I was working on synthesizing it. I was actually the first person, and maybe still the only person on Earth, to have synthesized and then crystallized allolactose. This was one of my publications. This is like the only chance I get to honk my own horn. But there it is. There's the structure of allolactose. Oh, we can look at it. Because once you get the crystal structure of it, you can take it and put it in a beam. So I sent this crystal off to the beam line at Berkeley, the high energy beam light source at Berkeley, and they can, when all of them are in a regular array, and you shoot a high power beam of light at it, you can get the exact X-ray crystal diffraction pattern from it, like Rosalind Franklin was doing with DNA. It was exactly the same thing. And then you can get, from that, you can get bond angles, bond lengths, et cetera, and I won't take you too far down the rabbit hole, but you can calculate some other parameters called scalar coupling things like NMR stuff and blah, blah, blah. It's research. I'll kind of leave it there. But you need the crystal structure to be able to do that. And you can only get those bond angles by sticking them in a cyclotron and hitting it with a high-power beam of light. There we go. All right. We'll talk about sucrose on Friday, and then we'll have lab. I have a period now. If anyone needs to talk to me, this is a good time.